As artificial intelligence technologies become more prevalent in our everyday lives, pharmaceutical companies are also recognizing the transformative potential and impact it can have on their sector and future. In 2021, the global AI and pharmaceutical market was valued at over $905 million, with projections estimating it to reach over $9 billion by 2030. The premise for using AI in drug discovery and development, eventually to helping pharma optimize their clinical trial processes and manufacturing of drugs, lie in the use of algorithms to uncover meaningful patterns from vast amounts of data, from the chemical structure of target compounds to patient information. By integrating AI's capabilities into the biopharma research workflow, companies can save time, deliver greater patient impact, and drive significant value for the healthcare industry. In this presentation, we explore how AI is making a profound impact in pharma drug discovery and clinical research. The process of developing a new drug usually takes 10 to 15 years or more, and roughly $1 to $2 billion to bring to market. However, advances in artificial intelligence open new possibilities for new treatments and cures to meet patient needs more rapidly and inexpensively. The first step of drug discovery is often to generate as many quality drug candidates as possible, as quickly, with the highest probability of a successful transition to clinical development. Historically, the discovery process has been a pass-fail funnel with challenges at every step. In other words, a tedious and inefficient process, as one project can begin with testing 200,000 to 10 million compounds for the initial screen. On average, only 1 in every 5,000 compounds that enter drug discovery becomes an improved drug. In the ideal future, most likely with the advances in artificial intelligence and machine learning, the discovery process should only promote compounds for testing that are relevant for targets, leading to a streamlined and effective process. AI's use cases across pharma's research value chain spans 5 steps: target identification, target validation, hit identification, lead optimization, and the preclinical phase. The first and foremost step in drug discovery, target identification, is the process of identifying the direct molecular targets of small molecules, such as nucleic acids and proteins, from a number of data sources. By mining and integrating the data, researchers identify specific genes, proteins, and pathways that play a key role in disease. Artificial intelligence revolutionizes this process by integrating gene networks, biochemical pathways, and cellular response data to automate insights gathering, uncover hidden patterns and relationships between the data, and ultimately increase the efficiency in which researchers identify potential targets. An impressive use case of AI in target identification is its ability to accelerate the generation of large molecule structures. Normally, determining the structure of large proteins would take significant time. However, with AI, the process can be accelerated by 100 times. Moreover, with the speed in which AI algorithms can comb through data and literature, it stands to shorten weeks of hypothesis generation research to minutes. Once researchers select their targets, they validate the initial hypothesis through testing antibody interactions and modulating the drug's affinity to the target by changing the molecular structure. Phenotypic models and cellular models are used to check if the target is valid, as well as find important markers related to the disease. Through computer vision technology, artificial intelligence accelerates the screening plate image analysis by up to 10 times. Moreover, some evidence points to computer vision technology enabled by convolutional neural networks as being more accurate than classical approaches. In fact, an AI program developed by Google DeepMind called AlphaFold 2 proved that it can generate 3D models of any known synthesized protein and protein-protein interactions in a few hours, compared to the usual need for 6 months. Next comes arguably the most critical step to identify how compounds interact with a fully validated target: hit identification. The goal of hit identification is to ensure that the compound has confirmed activity against a biological target. With computer vision technology and virtual screening, the automated image analysis enables automatic cellular assay analysis and molecular property prediction, standing to accelerate small molecule high-throughput screening by 30 to 50%. During lead generation and optimization, drug properties including potency, selectivity, solubility, or stability are analyzed and improved. Traditionally, the lead generation and optimization step of drug discovery is pursued through joint computational and experimental studies. Through the use of generative adversarial networks, a type of artificial intelligence consisting of two neural networks competing against each other to generate new data that resembles a known data distribution, rapid design iterations are created across small and large molecules. Biopharma leverage these generative machine learning models to optimize their most promising compounds and predict their efficacy, significantly increasing their library with 60,000 new compounds generated. Moreover, the informatics produce 100 times more experiments possible compared to previous screening. The preclinical phase, which usually takes 1.5 years and costs around $122 million, tests the drug in vivo, most commonly in mouse and rat research models, to ensure safety, learn the drug's metabolism, and predict pharmacokinetic interactions. With the use of artificial intelligence, patterns and correlations are identified to predict potential safety issues early on, helping researchers focus their efforts on developing drugs with a higher chance of success and fewer adverse effects. It also allows investigators to prioritize or deprioritize ongoing programs by identifying low-probability success programs early and reducing patient burden during clinical trials. Additionally, AI aids in generating hypothesis and potential dosage plans for adaptive trials. It can even leverage genomic data to prioritize indications based on specific genetic characteristics, tailoring treatment options to targeted populations, and identifying which patients are more likely to respond positively to the drug. As artificial intelligence continues to push the boundaries of drug development, many are convinced that the new wave of research and development for pharma and life sciences lies in AI. AI's ability to analyze vast amounts of data, uncover hidden patterns, and generate meaningful insights in a fraction of time previously thought necessary has significantly improved the efficiency and effectiveness of the drug discovery process. Over the years, we have experienced a rapid rise in partnerships between traditional biopharma companies and AI-driven drug companies. However, a significant concentration of funding and discovery occurs in smaller AI-driven players as biopharma companies continue to keep AI-driven initiatives away from day-to-day R&D efforts. AstraZeneca is an outlier to this trend. Known as early adopters of AI, AstraZeneca utilizes knowledge graphs and image analysis to gather new insights about diseases and identify biomarkers 30% faster than human pathologists. In fact, their long-standing collaboration with Benevolent AI enabled the identification of new targets in multiple therapeutic areas, including idiopathic pulmonary fibrosis. As we continue to explore the potential of AI in pharma, it is undoubtedly true that through the use of artificial intelligence, we can unlock even more possibilities in improving human health and well-being.